All right, next up from the Wargo lab, we have Hannah Brown. Hannah Brown is just beginning her graduate studies at North Carolina State University. She began volunteering in the Wargo lab at VIMS as a high school student and then went on to secure a full-time permanent laboratory technician position. During that time, she completed her undergraduate degree at Christopher Newport University. And today she's going to talk about implementation of digital PCR to quantify a novel viral shedding dose response. So Hannah, the floor is yours.
Awesome. Thanks, Matt, for that introduction. So today we're going to add on to Melina's study a little bit, but just to recap. So from Melina's presentation, we saw a host jump of IHNV from sockeye salmon into trout in the 1970s. And then she demonstrated that we've seen an increase in the virulence of IHNV over time, specifically within that newer group of isolates. And so this study is designed to kind of tease out how is virulence linked to this viral shedding, and is there a dose response linked to the shedding? So just to back up and make sure we're all on the same page, we'll define what shedding is. So in these experiments, we're exposing the trout to the virus in a bath immersion scenario. Infection should take over, and then the trout should shed the viral particles back into the water. That shedding, we're able to take a water sample of and then measure for both concentration of viral particles, which Melina touched on a little bit, but also just presence, absence of shedding in total. So if we relate that back to some other dose responses that you may be more familiar with, we have one that's typically studied, which would be an infectious dose 50, or the dose required to cause infection in 50% of the individuals. Another example would be a lethal dose 50, or the dose required to cause 50% of mortality within the fish. And so we looked at a completely novel dose response looking at shedding dose 50, or the dose that was required to induce shedding of 50% of the fish. So you can kind of compare it to infectious dose 50 of likely all of our fish had infection because they're shedding, but it could have taken a higher dose to get 50% of the fish shedding versus 50% of the fish infected. So in order to look at this, we needed a very sensitive molecular assay. And historically, we had been using quantitative PCR within the lab, and it was working great. But we wanted something that was a little more sensitive to tease out those positive negatives at really low doses of viral infection in the fish. So if we look at a comparison between digital PCR and quantitative PCR, we can see that digital PCR is based off an absolute quantification. So it does not require a standard curve to give you a quantity, where quantitative PCR is based off of relative quantification. So you have to have a standard curve that you're running on each and every plate to have something to refer back to determine the quantity of viral copies within that sample. Another difference is that digital PCR is an endpoint PCR. So much like a traditional PCR you would run out on a gel, you only get results at the end of the cycling conditions. Where quantitative PCR is giving you those CT values at each and every cycle. So it's measuring the amount of virus present or copies present at each and every cycle. But that allows the digital PCR to excel at the low copy numbers. So the particular platform that we chose was the QIAcuity platform. And so it's a nanoplate-based technology. So you take your master mix, take your DNA, mix it all together, and then it divides it up into the particular format we used was 8,500 little wells. So the odds that you get one copy in each of the wells or three copies in each of the wells, it uses Poisson statistics to form a normal distribution to decide how many copies are likely to be in each well. But because of that, we're able to detect if one of those 85,000 partitions is positive. Where qPCR, if you only had one copy, the machine has to read that in a larger well. And so it just doesn't excel as well at those smaller copy numbers. But it does do better at larger copy numbers where the digital can be oversaturated and just not give you the resolution you're looking for. Another benefit of the digital is that it's able to overcome inhibitors. So in the same way I described, it's taking the DNA or RNA and dividing it between all these little nanowells. It's also dividing the inhibitors. So if you do have inhibitors within your sample, it's able to spread that out into many different little reactions. And it doesn't affect the PCR reaction as much as it does with a quantitative PCR, where if you have inhibitors, it's inhibiting the entire reaction because it's all in one well together. So armed with this new technology, we went back to our research question of, is shedding a primary driver between virulence evolution? And so in order to test this, we set up an experiment using 9 of the isolates that Melina talked about in her earlier presentation. These are primarily M genogroup isolates, so after the host jump. We tested them at 4 different doses, ranging from 2 times 10 to the second PFU per mil and up to 2 times 10 to the fifth PFU per mil. We dosed 20 fish per tank. We left them in a bath immersion with the virus for an hour, flushed them for an hour, and then divided them up into 20 individual tanks. And then we held them static for 3 days. At the end of the 3rd day, we took a water sample to identify whether there is or is not shedding. And so then we took these water samples and we extracted the RNA using the TCAM machine that Andrew mentioned before. And then we performed a 1-step reverse transcription digital PCR assay using the QIAcuity platform. And this was the data that we got. So as you can see along the top here, there are the 9 different isolates. And so you can see they span the years from 1974, which I believe is one of the isolates, and then up to 2017. And so if you look generally across, let me orient you. So down here at the bottom, we have the 4 doses. So we have the lowest dose down here and then to the highest dose. And then on the y-axis, you have 0 fish shedding and then up to 20 fish shedding, which would be the maximum per treatment. So if you look, we can see this general trend of as the dose gets higher, the number of fish shedding gets higher, which is what we expected. But if you look across this top line here, we have much lower shedding than we do in the 2016 and 2017 isolates down here. So we can see that this amount of shedding is increasing across both dose, which we would expect, and time as we get to more recent years. So if we look at the SD50, so as a reminder, this is the dose required to have 50% of the fish shedding, not 50% infected. We can see that in the earlier isolates, we're requiring about 4.8 PFU per mil to have 50% of the fish shedding. But in the more recent isolates, the 2016, 2017 isolates, we're down to only needing about 4 PFU per mil. So we can see that as they're becoming more virulent, it's taking less and less to have more fish shedding. So this brings us to our conclusions. So digital PCR as a method allowed us the sensitivity that we needed to detect shedding at even low viral copies. So 1 to 2 copies in a sample at those really low dose of virus that we gave the fish was essential to have the resolution for this data that we needed. And then we can see that the SD50 continues to decrease over time, indicating an increased infectivity of the virus. So if we think about that from Melina's study, that as they're becoming more virulent and as they're shedding more in the aquaculture facilities, this is important to think about transmission as a primary driver of virulence. So if it's taking less virus to have more fish shedding, then there's more potential for transmission within these aquaculture facilities, and thus causing a bigger problem. So it's thinking about ways to cull the fish or preventative measures to try to keep this transmission down is important. And so thanking all of the lab members for all their help and getting me to where I am now, and all the fishies for helping us run all these great experiments. And with that, I will take any questions.
Excellent, Hannah. Very well done. Nice work. Please, if you'd like, unmute yourself and ask a question, and or go ahead, send me a message in the chat or in the chat. If you have any questions. Dave Groman, another question from Dave. He wants to know, do you know which tissues or areas on the fish the virus is shed?
So I might have to default to Andrew on this, but I think that we're thinking about it's being shed from the skin or the scales into the water primarily. But Andrew can correct me if I'm wrong on that.
Some of that, I mean, a lot of it's going to be through like feces and urine, and then as well, like through the gill tissues, anywhere that the capillaries are close to the surface. But yeah, the digestive tract, reproductive fluids, and then also the gills are kind of point about the fin bases too. You get entry and exit. So yeah.
I was curious, and I apologize if you covered it in your kind of pro and con comparison. In terms of like reaction sizes and reagent use and costs, how does digital PCR compare to your, kind of real-time PCR assays?
Yeah, so for us, the comparison of price ended up being about the same with
Okay.
the caveat that we converted a two-step qPCR. So we were doing the reverse transcription separately and then a qPCR. And so because with the digital we were able to do it in one step, we were able to save a little bit of money there. So if we'd had to keep it as a two-step, the digital would have been a little bit more costly. And another benefit to the digital is you can load a lot more of your sample. So of our extracted DNA, or RNA, we were able to add more than we could in the qPCR, which also helped boost our sensitivity and our ability to see those low positives.
Yeah, especially when you're dealing on, out towards the end of the cycle, 37,
Yeah.
38, 39 CQs, that I could see there being an advantage there. So
Yeah.
excellent. Mikolaj has a question. He's joining us from overseas. Mikolaj, good to see you,
Yeah.
bud.
Hi, can you hear me?
Yes.
Yep.
Oh, great. So you had the funky results in 90s, the isolates from 90s. There was one which was having very high shedding, and then one which was having a quite low shedding. How are you explaining them?
So we have here, you're referring to these three isolates in the middle.
Probably the next graph, it's showing it slightly better. Yeah, this one.
Oh, the dose response here. So that would be the 220/90 isolate. So as you can see here, there's much higher shedding for this particular isolate than the others. This might be a question for Melina, whether the 220/90 isolate was more virulent in your experiments as well. Because here we're seeing that there's more fish shedding overall at all doses for this isolate than the other two from 91.
But this virus,
2090 is one of our more virulent IHV
okay,
isolates.
That's what I thought, yeah.
okay. Thanks.
Mm-hmm.
Yeah, I think, you're going to get variation, right? And that's why we chose a lot of isolates, is because even within a year, you're going to have some variation. It does turn out that 220/90 is one of the more virulent ones. And so we're looking at these trends across time and averages, but there's going to be exceptions in there, right? And that's kind of the way nature works. And so why is there so much variation? Great question. We got to figure that out still. One we've wondered for a long time. Yeah.
Okay, thanks.
Yeah, we have one last question in the chat, and then we'll let you off the hook, Hannah. But are there any Azam asks, are there any genetic markers or specific mutations in the IH&V isolates that correlate with these decreasing SD50 values?
That is, I don't know that we've looked at that.
Yeah, that's a great question. We have a project right now that's trying to look at that, where we just sequenced 100 genomes, 100 isolate genomes, and trying to figure out if there's any correlates with virulence. And then Gail's also worked on like recombinant viruses to see if you can really like do the gene swapping. But at this point, we don't know what the genetic determinants of. We have some ideas, but we don't really know what the genetic determinants of virulence are.
Excellent. There's always more work to be done, right? So, Hannah, great talk. Thank you very much.